So I thought I'd make a video talking a little bit about the Omicron variant of SARS CoV2. And so right here I have an image, and there will be links in the description below for all the images and things I use. But if we look at this image here, what we see is that the Delta variant, which was the last one that became sort of the most propagated variant. It had 18 amino acid mutated residues in the spike protein, where the spike protein is the one that allows the virus to gain entry into human cells. 8 residues in the greater than 60% of available sequences. And so what that means, so when they talk about a variant of the virus, so a variant has a few specific mutations that sort of make it different from other viruses, but it can have a bunch of other mutations as well don't that don't occur in all of that virus, in all of that strain. And so when it says that there are 8 residues greater than 60% of available sequences, that means that those are the ones that are most prevalent in that strain. And so those are the ones that sort of, I guess, are the signature mutations of that strain. And so this is a structure of the spike protein for the Delta variant, and in red here are all the different mutations. So this little part highlighted in this kind of orangish color, that is the receptor binding domain. So that's actually the part of the spike protein that interacts with the human ACE2 receptor. And so mutations in that part are the ones that are very interesting or very concerning even to scientists. And we see here in the Omicron, we now have 43 amino acid mutated residues in the spike protein, and 34 are in the greater than 60% of available sequences. And we see a bunch of them here clustered in the receptor binding domain, which makes this sort of a very different sort of spike protein. It makes the Omicron variant quite different from other variants, including the Delta variant, and that's why scientists are very concerned about this particular variant here. This article is one that I'm going to be relying heavily on here, but I'll get more into that in a little bit. What I wanted to look at here, so this is the phylogenetic tree right here on this website. Once again, it'll be linked to in the description down below. We see here in green, these are all sequences for the Delta variant. We see down here in red, that is the Omicron variant, and we can actually look at this down here, and this goes from December of 2020, and we see that the beta and alpha variants were the big ones there. And then we see here the in green, this is where the Delta variant sort of came in, and we can see that the wider part here tells us that it is the greater percentage. So we see that this is the frequency, 100% at the top and 0% at the bottom of that particular variant. And so the Delta variant became sort of the big one here. But we see down here in red, this is the Omicron variant down here, and we can see that that is starting to grow, sort of similar to how the Delta variant was back here in April. And so that, tells us once again that this is a variant of concern. So this right here, once again, linked to in the description. So in the top part here, these are mutations that are shared by different variants. So we have here in the middle, this is the Omicron variant. And so these are all mutations shared by the Omicron variant. Down here are mutations not shared. And so we see this long list here for the Omicron variant, so that it has a lot more of these mutations. Uh, and so the S here on each of these little things is which protein. So S is spike protein. And then we have, for instance, the N440K. So that is an asparagine at the 440 position being mutated into a lysine. So you can actually mouse over it here, and it actually says mutation of amino acid 440 in spike protein from asparagine to lysine. And so those are the different mutations that show up in the Omicron variant. And you can also see the alpha, beta, gamma, the delta variant, the kappa, eta, iota, lambda, mu, and so forth. All right, and then so here, this gives a little bit about each of the mutations. And so a lot of this, so this has, you know, references to where the information came from. But I'm going to be reading from this article, and I actually copied that over to here so that I could highlight some things. So this is just a section of this article here, which will be linked to in full in the description down below. All right, so the more disturbing aspect of the Omicron variant is this large number of mutations, 50 in all, with 30 relating to the spike protein. Of those 30, 15 pertain to the receptor binding domain, a part of the spike area that is vital to begin invasion of cells. All right, so once again, that's just kind of what I was talking about before. There are a lot of mutations that we can see here in the spike protein and in the receptor binding domain of the spike protein. And I actually have, this is an actual structure of the spike protein, which I have down here in this sort of pinkish salmon color. And we can zoom in up here. And so I have highlighted here. So these two green residues, so one of them, this one here, is one of the mutations found in the Omicron variant. And this one right here is as well. So in cyan are all the residues that are known to be involved in the binding of the spike protein, which with what I have up here in yellow, that is the human ACE2 receptor. And so we can see where that binding occurs. There are also mutations involved. So down here, so this is the furin cleavage site, and there would actually be a loop of amino acids connecting these two parts that I have here highlighted in magenta, which contains the furin cleavage site, but that is a disordered region, and so it didn't actually show up in the structure. But yeah, this is sort of what the actual structure looks like of the spike protein down here. Then this yellow and cyan up here is the human ACE2 receptor. So up here in the sort of overlapping with the purple, so that is the receptor binding domain, which so I sort of got these structures to align by aligning the receptor binding domain, which was present in this human ACE2 receptor structure, with the spike protein structure. And so that is how I actually aligned those. That's why there's sort of that overlapping part right there. But anyway, once again, the Omicron has a bunch of these mutations in the receptor binding domain. All right, so we move down here. In the spike protein of the Omicron variant, there is a striking accumulation of mutations in the S1 domain, so in the RBD, the receptor binding domain. Multiple mutations in these regions could help the virus resist neutralization by vaccines and monoclonal antibodies. And so that means that what that's saying is that it could resist immune response. So neutralization by vaccines and monoclonal antibodies. So monoclonal antibodies would be, like the antibodies that your body makes, and vaccines would be, well, that'd also be antibodies that your body makes because of the vaccine. The monoclonal antibodies are used as like a treatment, like if you get the monoclonal antibody treatment for this. So this is saying that the virus could resist neutralization by these things. So down here, so second, the combination of substitutions, so lysine 417 to asparagine, serine 477 to asparagine. So this is, so I think that is glutamate or glutamine. I always forget the Q. I think it's glutamine to arginine, and then this asparagine to tyrosine here. And the Omicron variant's genome is also thought to, but not definitively known to, be able to add to the virus's ability to evade the immune system. So once again, making it more resistant to the immune system. All right, so down here, some in vitro studies have also found that the combination of spike protein mutations, Q498R and N501Y, could significantly increase the variant's binding affinity to the ACE2 receptor. And so the Q498R and the N501Y are the ones I was showing on here. So those are the ones that I have up here in green. So this is actually a structure of the non-mutated version. So these ones in green are the non-mutated ones, but those are the positions of those amino acid residues. And you can see that those are right here at the binding site to the human ACE2, and that's why those ones are of interest. And this is saying that that in vitro studies, so not just looking at the structure and saying, oh, that might be the case, but actual studies have also found that the combination of those two mutations, so this one, I believe, is new to the Omicron variant. This N501Y is found in other variants as well, but this is saying that the combination of the two could significantly increase the variant's binding affinity to the ACE2 receptor. So third, 4 new substitutions. So this Q339D, this S371L, these S373P, and this S375F could present additional obstacles to certain antibodies. A cluster of substitutions near the spike's furin cleavage site could enhance the virus's ability to be transmitted through more efficient cell entry. So these around 339, 370, those are near that furin cleavage site, which I showed down here. So if we look at this, so yeah, that is the furin cleavage site there. All right, so if we continue on here, so additionally, there is a 3 amino acid deletion in the non-spike region of the virus. Scientists have observed that this deletion in the alpha, beta, gamma, and lambda variants, and there has been some speculation that this deletion could help the virus with immune evasion, possibly by compromising cells' ability to degrade viral components. And so this is saying that it's a 3 amino acid deletion in the non-spike region of the virus. And it says the spike region because the SARS CoV2 is, well, it's one large genome, which we can see right here. And each part of that genome codes for different parts of the virus. And so this deletion here, this 3 amino acid deletion, is occurring in one of these parts, sort of outside the spike. So this right here is the spike protein, and we can actually see it over on this one as well. But it's saying that it is happening on a non-spike protein, and so that it is saying that this helps the virus with immune evasion. because it makes it so that the cell can't degrade those viral components as efficiently. All right, so this is why scientists have been saying that Omicron variant has a spike protein that is dramatically different. And you can see just from this how many of these red spots are on here telling us how different it is. So this is why scientists have been saying that the Omicron variant has a spike protein that is dramatically different from the one on which the COVID-19 vaccines are based, and the variant's overall mutational profile suggests a potentially significant transmission advantage, something that we may be beginning to see in the real world as the Omicron overtakes the Beta and Delta variants in different populations, which is what we were able to see here, where we can see this is starting to grow quite a bit down there. All right, so this was the next section of that article. So in Gauteng province in South Africa, the Omicron variant has been rapidly replacing the Delta variant, which suggests a transmission advantage, according to Christian Elthos, a computational epidemiologist at the University of Bern. The Omicron variant has an estimated growth advantage of 0.38 to 0.43 per day compared to Delta, which translates to a transmissibility of 280% of that of the Delta variant. Again, these are early estimates based on limited data. Assuming the same generation time, the transmission advantage could act on two levels: higher transmissibility and better immune evasion. So this is saying that that the transmissibility is 280% greater than that of the Delta variant, which is the sort of most popular one right now. But it's it's saying that these are estimates are based on limited data, so that could be inaccurate. There would be large error bars on that percentage there. So this said, other experts have found such a high transmission rate improbable. So the disagreeing experts believe that there may have been a substantial undetected transmission in a geographically isolated place for some months before the existence became apparent. And, I believe the place that they think it might have actually come from was some I think it was like Botswana, maybe. So not actually South Africa, but South Africa is sort of where it's spreading from. It came from a different country, and so that was where it had been undetected. It had undetected transmission for a while, and that was sort of the place where it was able to sort of incubate and generate all these different mutations. So this said, we don't understand the Omicron variant's actual threats. There is early evidence to suggest it presents a higher risk of reinfection compared to other highly transmissible variants. This means people who contracted COVID-19 and recovered could still catch it again. Yeah, and so that is pretty much, I mean, once again, as the article says quite often, this is all based on very early, preliminary, limited data, but these are all the reasons why this has become the variant of concern, or the VOC, as they like to call it. And so, yeah, we can see that this is quite a bit different from other variants, particularly this Delta variant, and so that gives us reason to be concerned about this. So I know, like the Pfizer CEO, for instance, has said that that the vaccine is still effective against Omicron. I wasn't able to really find any data on this, but we will have to wait and see in the future if the vaccines are still widely effective against the Omicron variant. But anyway, I hope you found this video helpful, and feel free to check out some of my other videos. I have a few others on COVID-19 if you're interested in that. But anyway, thanks for listening, and I will see you in another video.